Hello everyone, I'm Ling from the Laboratory of Nutrition and Food Hygiene of Harbin Medical University. The title of my presentation is The Effect and Mechanism of Vitamin K2 Supplementation on Type 2 Diabetes. Vitamin K is a derivative from the family of menaquinone with strong antioxidant biological activity of phytoquinone. The two types of vitamin K compounds most studied in human metabolism researches and applications are the naturally occurring vitamin K1 and vitamin K2. From a structural point of view, vitamin K1 has a phytoquinone side chain, while for vitamin K2, the third position is replaced by an isoprenoid side chain containing 4 to 13 units of isoprene, also referred to as MKN, where N represents the number of isoprene polymer unit. In terms of biodistribution, vitamin K1 is preferentially retained in the liver to assist in the carboxylation of coagulation factors. On the other hand, vitamin K2 is distributed into the circulatory system. One can expect vitamin K2 to have more functional roles in many extrahepatic tissues, such as the bones, vascular system, kidneys, intestines, pancreas, and genitals. Additionally, the vitamin K1 and vitamin K2 are found in different food sources. Vitamin K1 is mainly found in green vegetables and vegetable oils, while vitamin K2 is primarily synthesized endogenously by intestinal bacteria. The latter is also found in certain fermented foods, such as natto and cheese. Also, vitamin K2 has a higher bioavailability and has a longer half-life when compared to vitamin K1. Despite so, up until now, there is no recommended daily intake for vitamin K2. Only in terms of toxicity does the European Food Safety Authority recommends a daily intake of no more than 6 micrograms of vitamin K2 per kilo for adults. In children, a daily intake of 1.5 micrograms or less per kilogram was shown to not be associated with adverse effects. About its physiological function, the best known one is being a cofactor for vitamin K-dependent coagulation proteins. Vitamin K plays an important role in blood coagulation and prevention of excessive bleeding. Vitamin K1 is used commonly as a medicine. For example, 1 milligram of vitamin K1 administered to a newborn shortly after birth can prevent the occurrence of bleeding caused by vitamin K deficiency. On the other hand, vitamin K2 has the ability to promote bone formation, improve glucose and lipid metabolism, and ease vascular calcification. Its effects in promoting liver health, treating rheumatoid arthritis, and preventing Alzheimer's disease have also been recognized. Studies in recent years have shown that vitamin K2 plays a key role by acting as a cofactor for gamma-glutamyl carboxylase. Activation of gamma-glutamyl carboxylase in turn activates vitamin K-dependent proteins such as osteocalcin and matrix GLA proteins. This further promotes the carboxylation of undercarboxylated osteocalcin into its carboxylated form, and the same process for matrix GLA proteins. These two activated proteins then facilitate the transport of calcium from the blood into the bone, promoting bone mineralization and inhibiting soft tissue calcification in the process. This consequentially improves bone quality and reduces the risk of fracture. As shown in the diabetes map published by the International Diabetes Federal in 2017, in the Western Pacific region, where China is located, diabetes accounts for 1/3 of all global diabetics deaths. And it is estimated by 2045, the number of people with diabetes will reach 629 million worldwide. In the same time, the proportion of diabetes patients within the age group of 20 to 64 years old will increase, further demonstrating the seriousness of the problem. The human body contains a rich diversity of intestinal flora, and studies have shown that intestinal flora can influence diabetes. Furthermore, the literature suggests that as vitamin K2 originates from the intestinal flora, vitamin K2 supplementation can improve insulin resistance. With all that in perspective, we formulated the following hypothesis. Could vitamin K2 be used to improve diabetes by altering the intestinal flora? A study demonstrating the involvement of vitamin K2 in glycolysis in the intestinal flora seems to support our hypothesis. Based on the study background and assumptions listed here, we designed a randomized control trial. 102 diabetic patients were included and randomly divided into the vitamin K2 group and control group. The vitamin K2 group consumed 1 cup of 100-gram vitamin K2 fortified yogurt containing 60 micrograms of MK7 per day, while the control group consumed 1 cup of 100-gram non-vitamin K2 fortified yogurt per day. The intervention was carried out for 6 months with follow-ups every 3 months. At baseline assessment, follow-up visits, and at the end of the study, questionnaires and anthropometric measurements were performed. Laboratory tests for serum biochemical parameters, vitamin K2 intestinal flora tests, clinical tests including BAPWV, CIMT, and RMR were also conducted to investigate the effects of vitamin K2 supplementation on glucolipid metabolism and intestinal flora in type 2 diabetes patients. These are some of the photos from our field questionnaire, blood collection and laboratory testing sessions. Analysis of the data showed that the differences in baseline population characteristics between the intervention and control groups were not statistically significant. Using a linear mixed model, we performed 3 comprehensive blood measurements at baseline, at follow-up, and at the end of the study. Fasting insulin, insulin resistance, and the level of the phospho-uncarboxylated matrix GLA protein showed statistically significant reduction after the intervention, and the insulin secretion index was remarkably increased. Glycosylated hemoglobin significantly decreased by 0.86% in the intervention group, while in the control group, the value rose by 0.2%. In terms of fasting blood glucose, the intervention group decreased by 0.44 millimole per liter, while it is increased by 0.14 millimole per liter in the control group. Fasting blood glucose showed statistically significant decrease in the intervention group. Fasting insulin decreased by 3.53 microunit per milliliter in the intervention group. The control group showed a decrease of 1.44 microunit per milliliter, with a more significant decrease in the interventional group compared to the control group. Next, the HOMA-IS increased by 30.28 in the intervention group, and in the control group, the value increased by 17.37 as compared to the control group. On the other hand, HOMA-IR was reduced significantly by 0.43 in the intervention group and 0.19 in the control group. Compared to the control group, the reduction was statistically significant in the interventional group. For the phosphorylated uncarboxylated MGP, the measured concentration decreased by 140.24 picomole per liter in the interventional group, and by 58 in the control group. Compared to the control group, the reduction was statistically significant in the interventional group. The above changes in serum parameters indicate that vitamin K2 supplementation has beneficial effects on the various metabolic parameters of diabetes. Furthermore, through intestinal flora analysis, we found significant differences in the distribution and diversity of intestinal flora between the 2 groups after the intervention. The intestinal bacteria of vitamin K2 group was significantly more diverse than that of the control group. By PCA analysis, we found that there was no difference in terms of the distribution of intestinal flora between the 2 groups before the intervention. But after the intervention, a significant difference in the distribution of intestinal flora between the 2 groups was observed. Through screening of differential gut microbes between the groups, we identified 15 bacterial genera with different bacterial counts between them. We quantified more than 300 gut microbial host co-metabolites of the intestinal flora in fecal samples by using an ultra-performance liquid chromatography coupled to a tandem mass spectrometry. Using the OPLSDA analytical method, no difference in the composition of the co-metabolites between the two groups at baseline was found. However, at month 6, there was a significant difference in the composition of the co-metabolites between the two groups. Based on the multidimensional and one-dimensional assays, 21 potential differential metabolites were screened out. And the following conclusions were drawn using correlation analysis. We believe through intervention with vitamin K2, the intestinal flora homeostasis of the subjects were improved, which in turn led to changes in the gut microbial host co-metabolites, and ultimately improved glucose homeostasis in the subjects. This is the end of my presentation. Thank you very much for your attention.